Today on In Your Face Anatomy, we are dissecting a cow eyeball. Hey, cow eyeball, let me see. Show them your eye. Oh, yeah. We're going to get into that. All right, let's get into it. All right, let's dissect a cow eye. So initially, when you get your eye, it's going to look something like this, where it's got, you can clearly see the eye here, but we also have all of this muscle tissue and fat and connective tissue all the way around. So we're going to want to trim that off. But we want to be careful when we trim it off that we don't trim off, let me find it here, optic nerve. Just a little, oh, there it is. No, that's not it. There it is. That's my optic nerve in there. So we just want to be careful of it. So when we start cutting it away, where we're going to end up after we cut up all of the muscle is this. So we got all of the tissue taken off of it. And here on the end, you can see my optic nerve sticking out. It's blunt little structure. It's pretty thick though, so it's pretty easy to spot. So just, you don't want to cut that off. It's not a big deal if you do, but let's try not to. So now that we have the eye, with all of the extra tissue taken away, what we can notice is immediately we have my, that's what we want. See, that's a better version of the optic canal right there. Boom. So we got the eye. So all of this white part kind of going all the way around, this is my sclera all the way around, and then this is my cornea. The cornea is typically clear. It would be clear. The preservative we use dulls out the cornea as well as the lens. The lens is also typically clear and easy to see through. So this is our trimmed away eye, all nice and neat, looking good. Great optic nerve sticking up there, sending signals directly to the brain through the optic canal in the skull. So then we're going to make a cut. Grab my scissors, and we're just going to cut along that edge where my cornea meets my sclera. So remember, the sclera is the white of the eye. It is opaque, so light can't get through. The only way light can get into the eye is through the cornea itself. So if we open up the cornea here, make the cut, boom. Now we can see the iris of the eye. That's what we've got here. This black part that we see, the iris of the eye. Crap, let me grab a probe. I'm back. All right, so here's my probe. We got the iris of the eye, the black layer right here. Boom, all of this. And in the middle, there's the pupa, the pupil of the eye. I wish I had better light here. That's as good as it's going to get, though. So there is my pupil right in the middle. And so the pupil is really just a hole. It's also known as the iris aperture because of the iris. The iris is a ring of muscle here that can constrict and get smaller, letting less light into the eye if it's really bright outside. Or if it gets dark, the muscle will relax and make the pupil bigger, allowing more light into the eye. So that is our iris and our pupil, our cornea, and then the sclera is the white of the eye going around. Now, next up, let's do this one. All right. So I've got my cornea cut out. I take it off. I made a wider cut, just a little bit wider, taking off the entire protective layer, the whole retinal layer off of the eye. So we can still see the retina here, and there's the pupil underneath it. But if we move that out of the way, so I made another cut going around the iris, and get that off. And now we're going to get down into some of the funky, deeper structures of the eye. So the first thing we're going to see is the lens. So the lens typically, as I said, should be nice and clear. Crap. Let's dump the lens. There we go. So that is our vitreous humor. It's the fluid inside of the eye cavity itself. And then this is the lens. The lens is typically really soft and pliable, but now the preservative has made it firm. It's also typically opaque or clear, transparent, so we can see through it. And again, the preservative has made it solid and opaque, so light's not really going to pass through it. But the shape of the lens is important, which I'll get to in just a second. So the vitreous humor that we've dumped out is this gel-like substance that occupies the posterior portion of the eyeball itself. And it helps to hold my retina in place. So this is the retina along the back wall here. The retina is super thin and it's held in place by the pressure that's put up against it from that vitreous humor that almost looks like a breast implant down here in this gel-like substance. This is mostly just electrolytes and water. We also have another fluid in here. It's the watery substance on the bottom of the tray here. And that is an aqueous humor. So aqueous humor gets, it's nutritive, so it feeds a lot of this tissue in here. And then it gets drained out up close to the iris through what's known as Schlem's canal, or the canal of Schlem. But if blockage happens in that canal for drainage to occur, then that increases the intraocular pressure because we're still secreting this aqueous fluid, aqueous humor, into the eye cavity itself. And if we can't drain it out, then that pressure starts to build. And that's what leads to conditions like glaucoma. Now, along this back eye, I'm going to do this very gently. This is the retina. So you can see it's little wrinkles in it now at this point. It shouldn't have wrinkles. It should be nice and smooth. But we've taken out the humor, so now we don't have anything really holding it in place. So if I very gently start to peel back this retina. There we go. You know, I'm going to switch over and grab another eye where I've already done that. Boom. Okay. So now we can pull back the retina. There's the retina at the bottom. See how easily it pulls away from the edge of the eye. This is hard to do when you're looking at the camera. And it doesn't come completely out because it is connected to the optic nerve. There's some, there's some. So that was the retina that just flopped out. But here's the little piece of retina that attaches to the optic nerve going through the other side of the eye. There's my optic nerve. Boom. And you see that's directly connected to the retina. So that retina layer that we see here is still intact along the wall. It contains our rods and cones. So these are our photosensitive cells. So rods help us see in the dark. Cones help us see in bright light. And the pupil adjust the amount of light that gets into the eye to help control what's going on. Now, an interesting thing about the cow eyes, so this black part along the back is called the choroid. That's the vascular layer. It's got all the blood vessels and stuff in it to help feed and supply nutrients and oxygen to a lot of the tissues here, specifically to the sclera itself, but some of the other tissues inside the eye as well. And human eyes, the back here is almost all sclera. It's all black. But in prey animals like cows and sheep and goats, we have the tempora here. And this is this iridescent color. It's just really bright, really shiny. It's almost pretty in its construction here. And what it does is it reflects light back towards the lens. And this is what allows animals to see in the dark. And with that, I mean like nocturnal vision, being able to see at night. This is also what creates eye shine. So if you've shined a flashlight and you caught your cat's eye, reflects the light back. Cow eyes do that as well. Crocodile eyes or alligator eyes. There's lots of animals. Certain animal species have this if they need to see in the dark. So that is what causes eye shine. And we see it really in prey animals, but some predators too. The cats, for instance, of course, have that same kind of eye shine going on. All right, and that's pretty much it for the eye. So what we get is light coming in through the pupil. The arrangement of the cornea. Let me see if I can find a corneal piece. There's one. See if we can't pop it out on this side. Boom. Yeah, a little bit there. All right, so here we still have the lens attached. So this is cool. You see the iris going all the way around. Now, along the edge of the iris, what we're going to have here is called the ciliary bodies. Ciliary bodies are kind of an expansion of that choroid, that black area inside that we just talked about that humans have. So the ciliary body expands and it is an expansion of that choroid, and it has these coronary ligaments. So let me see if we can't find a coronary ligament in here. I'm trying to pop it over to see. I'm trying to get some light in there. You see it's being held up by these little tiny ligaments. Try my best. Try to get in the light a little better. Let's try folding it up. There we go. There we go. So you see that those little tiny ligament attachment points? If I mess with it too much, it's going to fall off. Those ligaments will break. We're holding it right in there. These little ligaments called our coronary ligaments that help hold the lens in place. There we can see them pretty good. Boom. So those are our coronary ligaments, and they help hold the lens in place. So the shape of the cornea, any light that comes in, is going to reflect that light towards the lens. The shape of the lens is going to reflect that light towards the back of the eye, and it's going to hit the retina. The retina has our photosensitive cells in it, the rods and the cones, so we can see better in the dark, or we can see better in bright light, just depending on our circumstances at the moment. In prey animals, like the cow, that light gets reflected back to the lens, so we get a second chance to absorb more light in the darkness, so we can see better, and that's what gives them the nocturnal vision. And because of the shape of the lens, when the image that we're looking at passes through the lens and gets projected to that back wall of the retina, the image there gets produced upside down on the retina itself. And then our photosensitive cells in there that make up the neural layer, what they're going to do is they're going to transcribe the image, the light that's hitting it, into electrical impulses that we can then send to the brain for it to interpret, so we can understand what we're looking at, so we can and see the world right side up, because the lens will project the image back here, but the image is upside down because of the shape of the lens. But our brain is able to restructure that to where it's right side up, so we understand what's going on. All right, that's the eye. Good luck.